from the Université de Québec à Montréal, and we will discuss about bedrest.
Hi, can you hear me? Yeah. Thank you very much for the organizing committee for inviting me and providing me the opportunity to present our work. It's going to load. Oh, it's my fault. Apologies for that. Technical issue. And thank you for David Hood for inviting me too. So today I'm going to present some data that were published, some that were not published yet, and we're pretty excited about the data we're collecting on the bedrest study that we recently conducted. So just to start with, we didn't hear a lot about muscle aging, so I'll spend a bit of time talking about muscle aging to introduce a few key concepts. So as you probably know with aging, it's not going very well for muscle. Just to illustrate this point, what you see on this slide here is on the left side here, you have an MRI of the thigh of a young individual, and on the right side, you get the MRI image of the thigh of an old individual. And what you can see is in this dark color here, this is the muscle. You can see that muscles atrophy with aging. You also see some fat infiltration that occurs with aging. And of course, if you lose muscle mass, you will of course lose muscle strength. So there is a decrease in muscle strength that develop with aging. And to make things even worse, whatever you're left with when you're old actually doesn't work as well when you were young. You have an impaired quality of contraction. So these three changes here, decreased muscle mass, force, and impaired quality of contraction, defines what we call sarcopenia. So today we'll focus a lot on mitochondria. I don't want to be misleading. Sarcopenia is a very complex and multifactorial process. But for the sake of time, I won't describe the whole slides. We are going to focus today on the role that mitochondrial dysfunction play in this process. So just to illustrate very briefly, I cannot go through the whole literature, but there is a consensus that is emerging now in the field, is that with aging, we have impaired mitochondrial energetics that develops, and we also get an impaired of the functioning of the mitochondrial permeability transition pore. This one is less covered, so I'm going to spend a bit of time on the mitochondrial permeability transition pore in the introduction. Because when it opens in skeletal muscle, it has the potential to trigger many relevant pathways for muscle mass regulation. And what I mean by that is, so this pore, by the way, is of unknown molecular compositions, or there is huge debates in the literature. The ATP synthase was implicated, the ANT was implicated, the phosphate transporter was implicated, cyclophilin D, etc., etc. Many, we don't know really what it's made of right now, but we know it can open. And when it can open, when the MPTP opens, well, you can release pro-apoptotic factors like endonuclease G that can translocate to the nuclei and cause apoptosis, which is not good for muscle. You can also activate caspase 3, which you probably know in the apoptotic pathway, but it was recently shown that caspase 3 can actually cleave some myofibrillar proteins, which is relevant for muscle atrophy. You also get, when you open the MPTP, a huge burst in ROS production, which can inhibit protein synthesis and trigger the atrophy program. So with the induction of MRF1 and natrogen1, for instance. So when this pore opens, you can really trigger many pathways that could regulate muscle mass. So on the protein synthesis side and protein degradation side. Just to show you some work that we've done in humans. So we've recruited very healthy, very active young and old individual in this study. They were training over 150 minutes per week. And we did MRI of the thigh to confirm that there was, if you look at the vastus lateralis cross-sectional area, that we could detect atrophy in our old group. And because MPTP, I didn't mention that, I need to mention that, you can easily test how MPTP functions, because one of the triggers for MPTP opening is calcium. So if you take a biopsy and you have your muscle fiber, you prepare it properly, you can challenge the mitochondria with calcium, and you can see how the mitochondria uptake the calcium and then release the calcium when the MPTP open. So that's what we've done here, and basically what you can see is that if you compare young and old individual, you get a decline in the mitochondrial calcium retention capacity, and you get what we call a sensitization of the pore. That's when you challenge with calcium, it takes less time for the MPTP to open. So you might be here, okay, that's an in vitro assay. Is the MPTP actually opening in vivo? What I've showed you in the previous slide is that if the MPTP opens, you can release pro-apoptotic factors that go to the nucleus, such as endonuclease G. So this is what we probed for in these biopsies. We did a stain in red for endonuclease G, and in blue for DAPI, and in grid, if my memory is not failing me right now, for dystrophin. So you can see the fibers here, and you can see with aging that there is an increase in the proportion of nuclei that stain in red. So this is nuclei positive for endonuclease G. You can see there is a threefold increase. So this is just to show you in humans, this pore very likely opens and very likely contributes to the muscle aging impairments that we see with aging. So, just to summarize here, so we're on point. We know that there is substantial literature now showing that mitochondrial dysfunction contribute to the muscle aging process. But to make things even more complicated with aging, when you want to study aging is, and this is a very nice review paper from Stuart Phillips and others, that basically, like the idea that I'm presenting you is kind of a linear decline in muscle mass and function, but this is not the case with aging. All the individual will have injuries, will have to be hospitalized, so they will have to undergo bed rest for several times. So basically what you see is that probably the normal, real evolution of an individual is that you have a progressive decline with aging, and on top of that you will get disuse-induced muscle atrophy that accelerate the dysfunctional impairment, the impairments that develops. So there is a substantial literature here, and I do not have the time to review it, so I will steal a paper from Scott Powers here, a very nice paper, that shows that mitochondrial dysfunction is a common denominator in aging and disuse-induced muscle atrophy. So again, if you have disuse-induced muscle, if you expose someone to disuse, you will get mitochondrial dysfunction. Another very important problem with the aging population is that the prevalence of obesity is on the rise, and the prevalence of obesity is extremely high in the old population. Same principle here, don't have time to review the whole literature, but although this field is more controversial, there is association between obesity and mitochondrial dysfunction in skeletal muscle. So basically, if you look at an old individual and at aging in humans, it's a very complicated process, and you can have obesity, aging itself, and disuse that can contribute to trigger mitochondrial dysfunction, which can lead to or contribute to the sarcopenic process. Now that I said that, I hope you will be sensitive that studying people that are old with obesity or that are old undergoing disuse is very important. So it was very nicely covered by previous speakers. This is going to help me save a lot of time, these slides. I'm showing you here what is called mitochondrial quality control processes, most of them. So what you have you may ask how a normal cell is actually maintaining mitochondrial health. And there are many processes that can do that. And I will simplify it today by breaking it down into three categories. The first one is mitochondrial biogenesis, creation of new mitochondria. We've heard a lot about this. PGC1 alpha is a major master regulator of mitochondrial biogenesis. We probed it in our end here because of antibody problems by probing for TFAM, which is a mitochondrial transcription factor, which is easy to detect. So this is what we've done. You also have mitochondrial dynamics, which was very nicely described before, so mitochondrial fusion and fission. What I'm going to talk about today in this talk is about MFN2, OPA1, and MFN1, which regulate mitochondrial fusion. And I will talk about GRP1, which regulates mitochondrial fission. And the last part is mitophagy, so the recycling of damaged mitochondria through the autophagy process, which is regulated by many proteins, including parkin. And Andrew got me scared this morning. I thought he was going to say that parkin is not involved in mitophagy with exercise. Thank you for saying it is. That would have made my talk very complicated right now. So I just want you to know that alteration in these processes were implicated in the aging-related loss of muscle mass. We've published a few papers on this topic. And I just want to highlight one of the papers we recently published that shows you that playing with these processes might have some relevance to fight muscle aging. And this paper that I mentioned is a paper in which we overexpress parkin in old animals. So we used adeno-associated virus to overexpress parkin, and we started with old animals at 18 months of age. Usually at 18 months of age, you don't have a lot of features of sarcopenia that are established yet. So we started at this point thinking if we overexpress parkin late in life, but before sarcopenia is there, can we alter the course of sarcopenia? Can we have some positive impact on the trajectory of muscle aging? So this is just to show you that we were successful in overexpressing parkin. On average, we basically got a 10-fold increase in parkin expression with our injection of adeno-associated virus. We also injected our control, so we had a control AAV we injected that expressed GFP. We injected that in the young animals. You can see That's the young animals here are here for the mass of the tibialis anterior. You can see the very nice effect of aging here with muscle atrophy that develops with aging. And when you overexpress parkin, you actually attenuate this aging-related loss of muscle mass to the point that you don't see a difference between the young group and the old group. You can see a representative image of how the muscle look at the end of the experiment. So we overexpressed parkin for 4 months, and we collected the muscle at 22 months of age, where we know atrophy is supposed to be detectable at the time. This is just to show that we confirm the same result at the fiber size in another muscle, in the gastrocnemius muscle. We also did some in situ contractility, because it's good to increase muscle mass, but if it's not functional, there is no point. So is the increase we get when we overexpress parkin functional? The answer is yes. You have the force frequency relationship here, and you can see that for any given stimulation frequency, we had more muscle force after parkin overexpression. And the last piece of data that I will feature from this paper is that with aging, you get an increase in the number of tunnel-positive myonuclei, which are, so tunnel is a stain that stains for myonuclei undergoing, or nuclei undergoing apoptosis. So you see the nice increase in apoptosis with aging, which was partly prevented by parkin overexpression. So this was just, the rest of my talk will be only in humans, but this was just to sensitize you to these pathways here. Just one example to show that if we play with this pathway, we might actually have a way to fight sarcopenia. So I will, I should say here, I will present 2 studies now. One that was published recently, and a second one that is not published, the one on bedrest. The first one, we studied the impact of exercise on many different things, but functional capacities, muscle force, muscle power, and processes regulating mitochondrial quality control in humans. The second study, I will describe it a bit later. I'll start with the first one. That was spearheaded by Vincent Marcangeli, which is present in the room. So if I don't answer all your questions, he's probably the guy to go to. So in this study, what we've done is that we recruited what I would describe as a whole lot of people. We had, you can see here the ratio man-woman. You can see that there are 2 groups. There was a placebo group, and we also had a group that had citrulline supplementation. Today, I won't have time to describe citrulline at all, but we studied it because there was preclinical data suggesting it could trigger lipolysis and have some positive impact on muscle mass in aged animals. So this is why we tried it. I'll show you a few results on citrulline, but if you have more questions, you can come to see me afterwards or read the paper that is indicated at the bottom. So basically, we recruited 45 participants in the high-intensity interval training group that received the placebo, and 36 in the group that received citrulline. So we had a decent amount of participants in this study. This is the protocol that I would describe very briefly for the sake of time. So basically, we had a lot of baseline tests. I won't detail all of them. I will just feature some of the key results of this study. So you can see we tested for functional capacities, muscle strength. I didn't put it here, but muscle power too. We did DEXA to assess body composition. We had some blood samples. I will not show any data on blood, but we do have papers in preparation with the blood data right now. And we did muscle biopsies in the vastus lateralis. We also did adipose tissue biopsies that I won't cover today. I will stay focused on the skeletal muscle. Then this is how our high-intensity interval training went. So because it was, just to feature, sorry, I forgot, that's the age of our participants. So pretty close to 70 in both groups. So really like what would be considered older adults in this case. So for older adults, you don't want to expose them to treadmill necessarily, especially, I forgot to mention, sorry to go back and forth, all of these participants were considered obese. So all men were above 27% in fat mass, and all women were above 40% in fat mass. So they matched criteria for obesity. So that's a study that was done in obese older adults. So just here, we did a study with a lot of obese older adults, so it's possible to do this, and I'll show you some cool results right after. So we did this training not on a treadmill, because that could have been problematic with the impact. We did it on an elliptical, just to make sure we lowered the impact as much as possible, and the enjoyment for the participants. So this training here, you can see that each session was 5-minute warm-up. Then we had 10 sprints that were repeated of 30 seconds. That was like really like high intensity in the way they were doing it. You can see on the board scale it was above 17. And then we had 1 minute and a half of active recovery. So they were basically like going easy. So That's only 10 times 30 seconds. That's basically the training. And then we had a 5-minute cool down. So you have the detail here of the supplementation. Again, that I won't have time to delve in detail. So these are the first results. So we studied them, for 12 weeks, and then we repeated all the tests post-intervention to see the impact of our intervention. So what you can see here is, I'll summarize for the effect of citrulline, so you guys are not actually perturbed by citrulline. There was zero effect of citrulline of these parameters. So basically, I can explain one group, and that's the same for the other one. So what you have here is the change in 6-minute walk test performance. So how far can they walk on a 6-minute walk test, which is a very, like in the clinics, it's really seen as a very good test to see if you improve functional capacity. And you can see here how the data are distributed. We got a significant improvement in the performance at the 6-minute walk test. We also did a step test where the participants basically have to alternate the steps on a step in front of them to see how many steps they can do in a given amount of time. You can see very, very nice improvement in both groups. So high-intensity interval training was very efficient here for the 6-minute wall test and the step test. You have a test here from the chair. So the lower your results at this test is, the best it is. So you can see a very nice improvement here. And same thing for the time up and go. So all of these tests are validated tests in the geriatric setting for older adults. So any improvement in this test is considered a gain in functional capacity. So very across the board, all tests that we've done, we improved the functional capacity of our obese older adult population, which in itself is already a pretty cool result. We then looked at what happened to lean mass using DEXA. And you can see here you have the total lean mass in both groups. And we looked at the leg lean mass to be a bit more precise in our assessment. Because the elliptical is mainly working the lower limb, although you still have your arm involved. It's mostly the lower part that is involved. And what you can see, again, same thing, no impact of citrulline, so don't be distracted by that. Both groups, what they show here is that, we improved, increased lean mass after 12 weeks of high-intensity interval training in this population. And we have a similar picture here for leg lean mass. You can appreciate, though, that there is different responses. Some people actually lose lean mass in our assessment, but in average, we got an increase in lean mass. Very interestingly, we also assessed lower limb power with the leg power rig, and what we found is that our training modality was efficient in increasing muscle power. So if you remember one thing here about this is that high-intensity interval training, if you can do it, if the population that is obese and old in front of you can do it, they should do it. That's basically what you can keep from this. The only slide on citrulline, and I have zero explanation for why we got these effects, is that we did see some differences between groups. Citrulline actually was the only group, so the group that took citrulline was the only one to actually show a decrease in fat mass after our intervention. Could be related to the lipolytic effects of citrulline. We have no data to suggest that, but that could. And if we look at the change in quadriceps strength, we did see a positive impact of citrulline supplementation, which actually this group increased more muscle strength after the training. So now, what we've done is that some participants were okay to actually undergo muscle biopsy of the vastus lateralis. You can see it's not our full sample size. That's the sample size we got, 13 in the first group and 14 in the second group. I will make the story very short for citrulline. There was zero impact of citrulline, which allowed us, again, to merge our group here to increase our statistical power. Because in humans, you get a lot of variability. And for everyone working with Western blots, you know you get a lot of variability. And the two combines are sometimes pretty challenging to work with. So we pulled all of our groups because we couldn't see anything from any effect of citrulline to just gain more power in our analysis. And first thing we did is to probe for TFAM, which is, again, a mitochondrial transcription factor. We upregulated when there is mitochondrial biogenesis. And you can see here, very, very nice when we pull off our data here, very nice impact of the high-intensity interval training, which increased TFAM pretty consistently when it comes to variability in humans. If we look at TOM20, it's actually pretty amazing. Almost all of the participants displayed an increase in TOM20 content, which is a marker of mitochondrial content. It's a protein of the outer membrane. When we looked at protein now regulating mitochondrial fusion, we were able to see an increase. So high-intensity interval training in obese older adults increased OPA1 content, increased MFN2 content, and increased MFN1 content. So we got an increase in all the proteins regulating, or the one that we probed for regulating mitochondrial fusion. When we looked at mitochondrial fission, we did not detect any effect of the training on DRP1 in this population, although you can appreciate how variable the data were here. So I would still be careful in saying that there is no impact on fission, but at least we couldn't detect it in this study. Last thing we did is that we probed for parkin content, and so now a marker of mitophagy, and what we found is that parkin content was increased following high-intensity interval training in our group. So just to summarize, because I want to spend some time on the last project that we've done, high-intensity interval training in obese older adults increased functional capacity, very efficient in increasing functional capacity, increased lean mass, increased muscle power, and so we don't have any more functional data, but it's very descriptive, but difficult to be more mechanistic in humans. We have data now showing that high-intensity interval training actually increases markers of mitochondrial content, biogenesis, dynamics, and mitophagy. So high-intensity interval training might be a way to, as was suggested in previous studies, to improve mitochondrial health in this population here, which is in dire need of improving mitochondrial health. So now I move on to the last part of my talk, which I think is even more interesting than the previous parts, which is a recent study that we completed on bedrest. And we had a countermeasure in our study, which was exercise. So very important here is that the participants stayed in bed all the time, and not we because I wasn't there, the group that actually trained these participants find a way to train them in bed with some modalities that could be implemented potentially in the clinic, okay? So this was funded by the Canadian Space Agency and CIHR. Important to mention here, for relevance for Canadian Space Agency, this bed rest was done with a tilt, so 6-degree tilt down, head down, just to mimic the blood distribution that happens when you go in space, okay? So there is relevance for space travel too in this study. So you can see here we had 11 participants in the control group, 11 in the exercise group. We had almost even number of males and females included, or men and women included in our study. You can see their characteristic here. So here they were a bit younger, but we didn't want to push too far because we know bed rest is bad for you. So it's still one of the rare studies that have focused on older adults and bed rest. This is the study protocol. So you can see they underwent a lot of tests, and probably this panel is not actually complete because there were 8 teams working on this, 8 teams across Canada working on these bed rest studies. So probably many more measures were done, but that's the one I'm aware of, and I will talk of some of them. So you can see we had 5 days to basically assess the participants. Then they underwent 14 days of bed rest, and we had a recovery of 7 days. I will talk about data on muscle biopsies at first day when they started the bed rest, put in bed biopsy, and then we go. Data points after 8 days of bed rest and 14 days. And I will show you data on the recovery at 7 days post bed rest. I'll directly dive into the countermeasure here for the sake of time. So every day they were doing 1 hour of exercise in 3 bouts. You can see if you look, there was some session of high intensity interval training, continuous aerobic training, and strength training. So alternate over the 14 days period, 1 hour a day. You can see some images here of the training, resistance training, different modalities. With the students and staff that train all of these participants. Of course, we took muscle biopsies. You can see Maud Dulac here, one of my students, and José's students, José Moret's students, having fun with the muscle biopsy samples. And what we've done is high-resolution respirofluorometry to assess mitochondrial respiration, H2O2 emission. And we did some quantification of markers of mitochondrial quality control with Western blots. So I'll start with bad news here, with the DEXA data, which we're not still sure to understand here. But basically, 14 days of bed rest, it's it's a decrease in leg lean mass. If you look at whole body mass, I think whole body lean mass, I think it's -3 to 4% over 14 days, and here you can see the data. We thought exercise would prevent this drop. This is not what we got. But I have good news. I have good news coming, I promise. 14 days of bed rest is a pretty big impact on VO2 max, which was measured. This decrease in VO2 max fully prevented by exercise in bed. Probably of a lot of interest for a lot of you here, this is an OGTT, or oral glucose tolerance test. So we challenged our participants with glucose. You can see both groups were comparable at baseline. This is what happens to the control group. You can see that they handle glucose less well. You can see that the curve is higher. So clearly we impaired their whole body glucose metabolism by 14 days. This is not new, that was reported many times. What is newer is that exercise had a very nice protective effect. It's not total. You can see there is a trend for an increase. So they did get worse if you look at the p-value of the ANOVA. They did get worse with glucose handling, but they got a significant protection when you compare the control and exercise after the 14 days of bed rest. So it was protective for glucose metabolism. Now, very interesting, mitochondrial respiration. I'll try to make it as simple as possible. We gave glutamate plus malate, which provides electron through complex 1 of the electron transfer chain. Then we gave ADP to test the maximal respiration. Then we gave succinates to give substrate to complex 2. And then we blocked mitochondrial respiration with, or inhibited the ATP synthase here with oligomycin and blocked the whole system with antimycin A. What you can see here, there is a lot of variability in the data, which is expected with humans. But what you have here is first day of bed rest, 8 days of bed rest, 14 days, and 7 days of recovery. I don't know if you can see this, but there is a trend for a decrease in mitochondrial respiration, which was reported, and it becomes significant after 7 days of recovery. So very important to keep in mind that if you have older adults in bed rest, you send them on the downward trajectory. It seems that they keep worsening, though the recovery was done with physiotherapy. It was a very organized program to bring them back to being functional after bed rest. If you look at the exercise group now, we did not just prevent the decline. We actually increased their respiration with the exercise training. If you look at what happens now to respiration with lipid substrate, we did not see a decline, so the ability to use lipids from mitochondria doesn't seem to be altered by bed rest, at least in our hands. But you can see the very nice improvement in respiration when you give lipids. So this is palmitate that we gave in this case. If you look at ROS production, complex data sets to analyze, but basically the bottom line is, in the control group, we saw a slight increase in H2O2 emission, which is in line with what was published before. Surprisingly, we also saw this trend of increased H2O2 emission in the exercise group. But remember, the exercising group increased respiration too. So likely increased mitochondrial content, which I will show you later. And what we could measure, because we measured H2O2 emission at the same time as H2O2 emission at the same time as oxygen consumption, we could calculate the free radical leak. How many electrons are leaking from the system? And you can see that bed rest in the control group increased electron leak, so you had a higher H2O2 generation per oxygen consumed, which was not seen at all in the exercise group. So some protection here in terms of H2O2 emission conferred by exercise. When it comes to protein involved in mitochondrial quality control, first thing we did is probe for 2 markers of mitochondrial content. TOM20, again, and VDAC this time, another protein of the mitochondrial outer membrane. We see a very similar, for the ease of comparison, now I'm comparing the control and the exercise group at the 4 time points that we studied. What you can see here is that there is a nice increase in mitochondrial content in the exercise group. And what the stats are telling us is that there is a trend for a decrease in the control group. Okay? So the control group is going down in terms of mitochondrial content. The exercise group is going up. So pretty nice protection in the exercise group. If we look now at protein involved in mitochondrial quality control, in dynamics and mitophagy, we could see some weird results that I'm not sure to fully understand right now, because we just got the data set very recently, so I'm still digesting a lot of it, and a lot of data are still coming every day from this study. So basically what you can see here is that there was one time point where we had a difference between the 2 groups. That was at the end of the bed rest, where there seemed to be some kind of protection conferred by exercise on MFN2 content. But one thing that I want you to appreciate is that for MFN2, DRP1, and Parkin, the changes with bed rest in the control group are not what we expected. We expected some kind of defects. We don't see a lot of changes with bed rest. There are some trends at some time points, but it's relatively modest, the impact. But what you can see is that exercise had a positive impact after 14 days of bed rest on MFN2. At 7 days post-recovery, we had a higher GRP1 content in the group that exercised versus the group that stayed sedentary, so just in bed. And we had higher Parkin 7 days post-recovery. So we will need to dig more into these data sets as we go, but what I want to leave you with, that's my last slides with content. To summarize this study, is that 14 days of bed rest in older adults is bad for you. It reduces lean mass. So it's pretty terrible, actually, when you think about older adults that are already not very good with glucose homeostasis. You reduce VO2 max, which is already low in this population, lower than young adult at least. You impair whole body glucose handling. You decrease mitochondrial respiration, even after recovery, like the trend keeps going after recovery, and you increase mitochondrial free radical leak. Not all, but a lot was prevented by exercise. I just want to stress that not all patients on bed rest can do the exercise we did, but I'm pretty sure that a lot of patients that are in bed rest can do some of the exercise we did. So we might have a way here to actually improve the health of patients by just allowing them to exercise in bed. This is a key message that I want you to leave with. And if we look at mitochondrial quality control, what the impact of exercise in bed rest, we increase mitochondrial biogenesis in content. We increase DRP1, MFN2, and Parkin, at least at some time points. So we need to dig more into what it actually means. But I want to thank all the funding agencies, all the students that were involved in these studies here. If you want to know more about mitochondrial calcium retention capacity, you can go see Marina at her poster here. And I want to thank all the PIs that were involved, and especially José Moret here, which is present in the room, and who is the PI that actually supervised and led the bed rest study. So if you have very technical questions about the bed rest study, you can ask José because he's the guy who actually led the whole work. Thank you very much for your attention.
Thank you, Gilles. I suggest that we take time for a short question. Zane?
Very short question. So the Parkin, in my understanding, is like regulated by PINK, and Parkin normally is being degraded, and so I kept seeing that Parkin as a marker of mitophagy, and then overexpression of Parkin. So I'm a little bit, I want to get a consensus here from the community, what we do with the data of Parkin.
So I'm not sure what your question actually, if I understood it exactly and precisely, but what was shown in cardiomyocytes is that PINK1 is dispensable for Parkin to induce mitophagy, which we don't know how, but it still occurs in the absence of PINK1, and it's mediated by Parkin. So there might be a PINK1 independent pathway of mitophagy. I don't want to confuse anyone here, but data from our lab now, which I couldn't present, is suggesting that Parkin does a whole lot of things than just mitophagy too. And I will give a very brief example of that. Is one of the very robust features we see when we overexpress Parkin is an increase in mitochondrial content. And the initial study that was done, I think, in flies, where I think it was, I can't remember the name of the Rana was the first author on this paper. It was a PNAS in 2013, I think. They have overexpressed Parkin in H flies, and they showed that overexpression of Parkin was sufficient to increase lifespan. And what they show, one of their key results, is that overexpressing Parkin increases mitochondrial enzyme activities, which is in line with what we see. So it seems that if you overexpress Parkin, you might play with mitophagy, which is the main described function of Parkin, but you somehow also trigger mitochondrial biogenesis. So we can discuss more if you want, but
sure.
It seems that Parkin regulates a whole lot of things other than mitophagy, most likely.
Okay, thank you. Thank you, Gilles.
Pleasure.
Yes. We'll close here this session. Thank you for attending it. We have a coffee break, but don't forget to visit poster.